Thank you. Thank you for that introduction, Sarah. I did my thesis research on glyphosate and AMPA accumulation in Camassia Quamash bulbs of Coast Salish Prairies. Camassia Quamash, both parts of the scientific name come from the indigenous words for camas, which is a culturally and ecologically significant plant in the lily family. It is a perennial and can live for 30 or more years. Here you'll see a Coast Salish prairie with Camassia Quamash flowers in bloom and an illustration of the plant with images of bulbs next to it. The bulbs are traditionally eaten in great quantity as a staple source of carbohydrates and were traded across tribal nations. To the left of the illustration, you can see what the layered bulb looks like when it is harvested, and to the right, what the edible part of the bulb looks like once its tunic is removed. This is the part that I sampled, and I will be referring to Camassia Quamash as camas throughout this presentation. Here you can see camas's range. The area that we are in is characterized by an oak and camas prairie ecosystem. The northern section is the Coast Salish Prairie region. The extent which I studied is highlighted in red. It is quite minimal compared to the camas's extensive range, which spans many ecosystems, altitudes, and climates. Wherever camas grows, indigenous peoples likely had a hand in sustaining the population. With traditional tending practices and reciprocal relationships since time immemorial. Camas's habitat is scarred and dwindling. Fields like those pictured here were once full of blooms to the extent that during the blooming season, the prairies looked like ponds of water due to the density of blue flowers. The map above shows the pre-colonial extent of the prairies, and the map below shows the current. There are many reasons for the deterioration of these prairies, such as land use change, exclusion of indigenous tending practices, and opportunistic non-native species taking hold. Those species, also known as invasive species, are at the forefront of restoration priorities today. All right, so to combat invasive species and make space for natives to thrive, herbicides are often used. Glyphosate-based herbicides are the #1 herbicide used in restoration. Glyphosate is the main ingredient in Roundup, which is a pre-formulated herbicide, and Roundup is not the only glyphosate-based herbicide used on prairies. Often glyphosate itself is mixed with other chemicals and applied. The herbicides are used in tandem with other restoration techniques. Common practice in oak camas prairies is to do a control burn, then spray with glyphosate, and then seed with native plant seeds. Here you can see an image of camas with the invasive species Scotch broom encroaching. The prairie images that I showed earlier are from a site that was once covered in Scotch broom, and glyphosate-based herbicides had a hand in getting rid of the bulk of it. So glyphosate kills through disrupting the shikime pathway, which is essential to producing amino acids in plants. In plants and soils, it is mineralized by bacteria into aminomethyl phosphonic acid, or AMPA, which is the other compound that this study is focused on. Glyphosate can enter non-target plants during overspray. It can kill plants, harm them, and accumulate in them. The image on the bottom right corner shows one of the methods of spraying, which is broadcast boom spray. Broadcast spray was the method used in this study. All right, so glyphosate enters non-target plants like camas in 2 ways: when the target plant is missed or through root uptake from the soil. Glyphosate enters the soil by hitting the ground directly through root exudates from sprayed plants and through dying plants litter. Once it is in the soil, it can be mineralized into AMPA, immobilized when it attaches to soil particles, or leached into groundwater. If it remains in soil, it can be bioavailable to plants like Camas, and AMPA is also bioavailable. You can see here the soil and weather and time characteristics that may impact how long glyphosate and AMPA remain in soil. The bolded factors are the ones that I accounted for in my research. Typically, the more clay, organic matter, and iron in soils, as well as the higher cation exchange capacity and lower pH the soil has, the longer the compounds remain. They tend to dissipate from soils that are high in phosphorus and well drained. More precipitation contributes to more of the compounds leaching from the soil, and both glyphosate and AMPA dissipate from soil over time. My research focused on the length of time between when an area is sprayed with glyphosate and when AMPA may no longer be in Camas bulbs, or glyphosate and AMPA may no longer be in Camas bulbs. There are very few studies observing glyphosate and AMPA uptake in herbaceous perennial plants like Camas. Those that do are in forests and show that compounds can remain in plants' roots for 12 or more years. So it follows that Camas bulbs would be at risk of glyphosate and AMPA accumulation. So if either glyphosate or AMPA are accumulating in the bulbs, there are some major health concerns. The way that glyphosate kills plants is through the shikimate pathway, which does not exist in animals, but does exist in bacteria and fungi. With the relatively recent recognition that bacteria is of huge importance to the human gut microbiome, research within the past 6 years has started to look at how glyphosate and AMPA may impact this bacteria. In vivo studies have not yet been done in humans, but they have been done in mice. And they show generational health impacts that include suppressed immune systems, being prone to asthma, metabolic dysfunction, including diabetes, behavioral irregularities, and gut bacteria dysbiosis. The species of bacteria that are altered in mice are also in the human gut microbiome. All right, so the question is, how does the legacy of glyphosate application affect glyphosate and AMPA accumulation in Camas bulbs? I chose 6 sites. To answer this question, I chose 6 sites, 3 that had been exposed to a glyphosate-based herbicide and 3 that had not. 4 of those 6 sites are on this map. The Chehalis tribe manages 2 of the sites and requested that the locations not be shared. I selected sites with cultural significance and traditional harvest in mind. And as glyphosate can travel through air during adjacent treatments, I also selected sites that are at low risk of being exposed to glyphosate by air drift. I did this through a GIS analysis that I will show later in the presentation. All sites were treated during winter months. Marion Prairie was treated in late December 2021, meaning that there was a 17-month time span between treatment and sampling. Scatter Creek and West Rocky were treated in January 2023, resulting in a 5-month and 4.5-month span. I want to note here too the different concentrations of glyphosate that were applied. Marion Prairie had a 2.5% concentration treatment, and Scatter Creek and West Rocky had a 0.96 treatment. At each site, I designated a 5 1-meter by 1-meter sample plots and extracted 8 Camas bulbs and 1 soil core from each. This totaled 30 composite bulbs and 30 soil samples. My plots were at least 30 meters apart, and I made sure to sample bulbs from a variety of ages of plants. I did this through, I determined this by looking at the number of leaves and if the flowering stem was present. I gathered data related to Camas bulbs, soil, and weather. I removed soil and tunics from bulbs to mimic how the bulbs would be consumed. I weighed them and then sent them to an offsite lab to be tested for glyphosate and AMPA. I did soil analysis myself, finding pH level, clay content, and organic matter content for each sample. I also used the USDA's Web Soil Survey to gather additional soil characteristics. Weather data came from the Olympia Airport Weather Station, which is the closest station to all sites. These are the results from my air drift analysis. Again, only 4 sites are pictured here at request of the Chehalis tribe. For the GIS analysis, I accounted for drift from roadside, commercial farm, and commercial tree farm application. I created a distance-based scale from very high to very low. All sites except Marion Prairie were found to have no risk of glyphosate air drift exposure. Marion Prairie samples were taken from within 50 to 100 feet of the road and had a low risk. My results. I sampled the top 10 centimeters of soil, which is the extent of soil that Camas bulbs inhabit. I found values that represent low pH, very low clay content, and high organic matter. With the low pH and high organic matter, you might expect that glyphosate and AMPA would be immobilized in soils. All of my soil results aligned with those from USDA research and other studies on these prairies. When I looked at other available soil characteristics, I found that they were somewhat excessively drained, sandy skeletal, and of andisols and inceptisols soil orders. The high drainage of soil and the sandy skeletal structure suggests that glyphosate and AMPA may be mobile in soils. So when looking at weather patterns, I found that all treatments were during consistently rainy periods. This graph shows monthly precipitation data. The x-axis shows months, and site treatment dates are indicated by the labeled white lines. The left y-axis shows total monthly precipitation in inches, represented by the dark line. The right y-axis shows the percentage of days in a month with some amount of precipitation, and those are represented by the bars. Studies in well-drained soil show that glyphosate and AMPA dissipation correlates with amount and consistency of rain. There were unequal amounts of precipitation between sites during the time between treatment and sampling, which indicates that there may be site differences in bulb accumulation. Here we're looking at a graph depicting ideal mineralization temperatures. More mineralization of glyphosate in soil means that less of it is available to plants and potentially more AMPA is available. Due to a combination of a couple of different studies of glyphosate mineralization, I determined that in Coast Salish Prairies, ambient air temperatures above 41 degrees Fahrenheit correlates to the most ideal soil temperatures for mineralization. On this graph, the x-axis shows months. Monthly temperatures are on the left y-axis and depicted by the solid line. Any portion of the line in the shaded area is under 41 degrees. The right y-axis shows the percentage of days in a given month where the average was above 41. Based on this data, there were 10 months of ideal 10 months of ideal mineralization temperatures for Marion Prairie, and Scatter Creek had 2 months and West Rocky had 1.75. The differences in prime mineralization months suggest that there will be variations in glyphosate and AMPA accumulation between sites. All right, so despite the soil, precipitation, and temperature results, as well as previous studies, there was no detectable glyphosate or AMPA in any Camas bulbs treated or untreated. This was found 4.5 months after a 0.96% glyphosate treatment and 17 months after a 2.5% glyphosate treatment. It's important to note that the results are highly specific to the small region of prairies that I sampled. Implications. I found there's no risk of glyphosate or AMPA consumption from Camas bulbs 4.5 months after a 0.96% or less glyphosate treatment and 17 months after a 2.5% or less glyphosate treatment. These results are quite encouraging, but should be treated with caution. They are highly specific to the soil characteristics of prairies surrounding Olympia, Washington, the climate of Coast Salish Prairies, and the herbicide application used in the studies. My hypothesis is that soil characteristics such as low clay content, excessive drainage, and sandy soils led to glyphosate and AMPA not being immobilized in soils and instead being available to dissipate. Climate and weather then likely played a role with mild, wet winters and consistent rain. Mild winters may have meant that soil was not too cold for glyphosate to be mineralized and become less available to plants. Consistent rain means that the compounds could be leached from the soil. Application timing is significant here. Herbicides were applied during the rainy season, meaning that immobilized glyphosate and AMPA could have both quickly leached from the excessively drained soils. Camas is also dormant during the winter, and the plant was not directly exposed to the herbicide. Additionally, as Camas is in the top 10 centimeters of soil, there's a question as to if glyphosate and AMPA leached out of reach of Camas's roots and is still available to other plants with deeper roots. So to my knowledge, this is the first study that analyzes accumulation of glyphosate and AMPA in Camas bulbs, as well as the first study to analyze the accumulation of the compounds in any prairie plants. The study provides great baseline information, but is specific to its conditions. I'm showing the map on the right again, just to highlight how small of an area my research covered. So exploring other climates and soils is super important, and this information should not be extrapolated to mean that bulbs are safe to eat in other regions. Research has also shown that different species in the same family don't concentrate glyphosate and AMPA in the same to the same extent. So this research cannot be extrapolated to mean that sister species like great camas can be safely eaten. All herbicides are different, and herbicides that are not glyphosate based are used on prairies as well. So we need to understand if these herbicides are accumulated in bulbs. Additionally, the concentration of glyphosate used in the 4.5-month application is on the low side of typical restoration applications. So we need to look at what happens with a higher percentage. We also need to understand the extent to which application timing played a role in the results. Finally, there are many more culturally significant plants in these prairies, and one is pictured here. The yellow flower next to Camas belongs to biscuit root. Thank you all for listening and for being here. I want to acknowledge Camas and Coast Salish Prairies, my ancestors who tended the prairies since time immemorial, the Chehalis tribe who I partnered with on this project, my thesis reader, and my support network. Any questions out there?